Hi everybody, I'm Tommy Temple. I'm the President and CEO of Grann Pharmaceuticals Inc. We specialize in mRNA solid lipid nanoparticle technology for the purpose of protein replacement therapy. Fair heads up to everybody who was here yesterday, I am going to recover some things, so if you were here, you're going to hear it again. But for everybody who wasn't and meeting me for the first time, that's who I am. So let's go into why we're doing this. Why does my company exist and all of that? So the name is a little strange. It's called Grann. That's named after my grandmother, who everybody in the company actually knew and loved, and we all called her Gran Ann. So it's named after her. Everything's dedicated in her honor, and also to everybody we've lost. So and as I go more through the presentation, I am a for-profit entity, but we really put a large focus on rare disease at cost to just try and help people as best we can. So what is an mRNA solid lipid nanoparticle? I'm sure everybody here, as a scientist, fairly familiar with mRNA, maybe not super familiar with solid lipid nanoparticle technology, and without going into anything super proprietary for us, from the mRNA creation side, we do everything in vitro transcription using DNA, which we synthesize. We do a lot of things differently that if you're really interested, I can sign an NDA with you and go into more depth on. But from that, we then go to our nanoparticle delivery system. We tag it with a lot of different things, again, which I have to go in more in depth on under non-disclosure. But essentially, to create a solid lipid nanoparticle takes 4 primary components, which is an ionizable lipid, DSPC, PEG lipid, and cholesterol. That's all mixed together with the mRNA using hydropholytic mixing. And basically, through hydrophobic forces, we're able to get the particle to form around the mRNA and create a little nanoparticle that's, for us, about 70 nanometers in size. From that, the entire goal is to target it to particular cells to express the healthy version of any given protein. And the big thing that has helped us a lot through FDA and going to clinic is the toxicology side of our particles is incredibly favorable. So the side effects that we see are very close to nothing, so far anyway, mainly because every component that we use degrades within the human body. The other big thing here with the mRNA is we do not modify our mRNA. Our goal is not to get the mRNA to stick around as long as possible. We envision doing multiple doses, mainly because, again, we don't want to elicit any kind of an immune response or inflammatory response. So we do not modify. That's a much larger conversation. If you want to pull me aside, happy to go more in depth on that as well. So let's go into treating. Oh, that went too far. Okay, there we go. So in terms of treating GABA-A variants, our focus is going to be on GABRA1 and GABRG2 originally. Obviously, the idea with the particles works incredibly well with loss of function. We have some data internally that also shows for gain of function, primarily by outcompeting the mutated version of the protein for membrane space. Now, I will readily admit that the reason that data isn't on here is that is also for a separate project that hasn't been done directly with GABA. That's what we're hoping to do in 2025. But essentially, by re-expressing the healthy version of a particular protein, you outcompete the mutated form for membrane space. And that involved a lot of patch clamp work with UC Irvine. I am not a patch clamp expert. Those questions will also go to our UC Irvine partners. So before I go into lots of details scientifically on everything, I want to give a big thank you to the El Nacave family. They've supported our GABRA1 and GABRG2 work so far, and the therapeutics are sitting in my fridge back in San Diego. So we would like to rapidly put them through preclinical trials and get to the clinic, and our vision is in 2026. So let's go a little more detail just on the mRNA and solid lipid nanoparticle side of things. A big part of this presentation is also going to be FDA oriented for how we go through that process at a fast speed. So let's first go into how do you measure solid lipid nanoparticles? Well, the primary thing that you do that with is dynamic light scattering. So dynamic light scattering uses Brownian motion to essentially give you particle size. Now, that measurement goes into an awful lot of physics. But essentially what you're looking for clean particles is, if you see those peaks over there on the right, that's 3 separate peaks. They all basically need to line up right over each other. DLS is used as part of the good manufacturing practices for Pfizer and Moderna in order to clear the COVID vaccines to go out. Because essentially, if you have any kind of particle contamination outside of doing sterility and bioburden testing as well, it's going to show up in that. You have a dust particle, those readings are going to get thrown way off. You're not going to get 3 clean peaks. And also, all 3 of your readings need to be within about a nanometer of each other, which these are. So this is a perfect particle creation, essentially. This has encapsulated mRNA. Just for reference here, for more details, if you were to encapsulate empty lipid nanoparticles, the particle size is going to go down to like 45 to 50. So you can also tell encapsulation percentage to a little bit. There's also a more in-depth test of how you actually see your encapsulation efficiency. We average about 90 to 95% of mRNA encapsulation. We'll see if the clicker works. There we go. I'm sure everybody's seen this a million times. Agarose gel images, tons of fun. But just to show that we have very purified DNA and mRNA. You have the DNA on the right. We're looking at about 17, 16 base pairs for GABRA1. And then for the mRNA agarose gel, you see that over there. It's roughly the same thing. Also, we sequence all of this every single time to release a batch. That's also required under FDA. So then let's go into more detail, and I'm going to break each one of these down scientifically on what we're planning to do. Before I get into that, I wanted to put this out here that we are experts in mRNA solid lipid nanoparticle technology, and we have some knowledge on GABA-A, but I very much want input from the scientific community on the efficacy studies that we're going to run. Toxicology and biodistribution and that kind of stuff we've done many, many times, but efficacy in particular, I want opinions. So please feel free to approach me afterwards. So for efficacy, what we've done in the past for other rare diseases is we choose particular mouse models and we do an enormous amount of testing within about 6 months. From the variants that we're focusing on, I know GABRA1, the mouse model that Dr. King has showed us previously, has a lot of things that we can measure, but for GABRG2, the model is fairly healthy, so there's some things we're going to have to go into there. But that we predict that takes about 6 months. That's what we've done previously. For toxicology, what FDA requires of us is an enormous cytokine panel, which I'm also going to go into. It's about 25 different cytokines that you need to look at to make sure you're not getting a massive inflammatory response to anything. This is particularly required in the efficacy testing. If something happens to one of the mice, I need to run a massive cytokine panel post-mortem to make sure I'm not having any nightmare scenarios occur in those mice. In terms of biodistribution, basically we're looking at expression of the protein, and I know it's strange up there. I'm listing an awful lot of organs. FDA requires me to do that. Everything is primarily targeted to the CNS for us for this particular particle, but I have to test everything because they want to know whether expression in various organs can cause any problems. And then pharmacokinetics. Pharmacokinetics we have run multiple times as well, but pharmacokinetics we're looking at injecting a given dose size and then we're measuring PEG, polyethylene glycol via an ELISA. So that allows us to determine how long the particles are in there. We use polyethylene glycol because the half-life of it is incredibly short. So once it's no longer part of the particle, it will degrade between 1 and 4 hours. A lot of the other pieces can take a little longer, so that gives us the best insight into how long the particles are in the bloodstream for. Now, realistically, if we used a very similar particle to what we've used in previous clinical trials, they allow us to reuse that data. However, since we're going to be tagging the surface of the lipid nanoparticle with something different, I'm going to repeat it. So going back into more details on efficacy testing, and I'm not going to read out the entire model strain. It's right there. The presentation will be out here. But those are the 2 particular models we're looking at for GABRG2 and GABRA1. Again, very open to input on that. If there are different models that everybody would like to see or multiple models, happy to do that. Same with GABRA1. And then, yeah, community input. So we went over that. Toxicology, there is the cytokine panel list that we have to use. That all goes through IDEX laboratories. So essentially we collect serum from these mice and send out an enormous panel to IDEX. This is what FDA has previously accepted from us scientifically to be able to proceed to clinic. So that's what we'll be using there. Let me click Biodistribution study, as I went over previously, we're looking at about 15 time points from 1 hour to 168 hours over 7 days. And we collect gonads, liver, brain, spleen, pancreas, kidneys, heart, and lungs. And what they have recommended us to do previously is ELISA testing as well. So essentially denaturing the proteins in serum and then running them through ELISA, get a protein expression level at every organ at a enormous amount of time points. That really allows us to get the dosing regimen correct, which is one of the biggest things that FDA requires of you. So pharmacokinetics, as I went through before, best measure is looking at DMG PEG 2000, which is the particular PEG that we use. That's a 30-day study. It involves 60 mice at 2 at each time point, and it has to be repeated. So that's probably the largest study in terms of mice that I have to use. Luckily for us, they are lenient on what model we use here because pharmacokinetics is not going to necessarily change in uptake. So they're basically okay with using a wild type mouse for the pharmacokinetic studies. So previously we used bulb Cs as our wild type. If there's a preferred wild type, that's fine with us. There we go. So FDA timeline for reaching clinic. This is what we've done previously to get into clinic, which will be starting for us in 2025. But obviously you need good manufacturing practices. So you need to have a full-scale laboratory setup that can operate under GMP. For us, we have that. We have a large partnership with Cal State San Marcos, which allows us to create therapeutics under good manufacturing. That's the first thing that FDA is going to hit you in the face with is where are you making this? And frankly, most of the largest arguments that happen in those meetings have to do with good manufacturing practices. Since we have that already, the first thing that you're going to do, which can be done while you're doing the efficacy testing, is to get a pre-IND submission in as soon as possible. So a pre-IND submission is going to allow you to talk to FDA and get their opinions on everything that you're doing. So this is really your first step, your first contact. Anybody who's doing anything, regardless of what I'm doing, needs to be doing this. The pre-IND submission, they'll tell you everything. You present your entire plan, you give them the idea of your clinical plan, and ideally they come back with thumbs up, thumbs down. They tell you to change things. And most of the time they're open to meeting past just the initial pre-IND. Because a pre-IND, they have a set time frame they have to respond to you in legally. And after you talk to them, technically they're not allowed to talk to you again. But if you ask very nicely, most of the time they're willing to schedule another meeting, which has been very beneficial. We've been able to meet with them multiple times prior to our most recent IND submission. After that, you need to determine treatment location. So a big thing that they ask for in the IND is where is this going to be injected, as in a place on the planet, hospital, where, what state, that kind of thing. And you obviously need to base that around the patients. So between your pre-IND and IND submission and what we're doing is you have to determine location and the patients that you're going to be using. They're going to ask for that specifically in the IND submission. So the IND submission is a bulky 12 to 1,500 page document that's going to include everything that you've ever done relating to the particle, all of your GMP manufacturing specs, all of your efficacy data, and I mean everything. Every picture that you ever took, every study protocol, it's the appendix, let's just say, can sometimes be 700 pages by itself of just images. So while you're doing the IND, the IRB can happen before or after depending on when your clinical protocol is created. So for the IRB approval, the Institutional Review Board, if you're a private entity like me, you can go to a private institutional review board. If you're working through an institution, obviously you're going to have to use your own IRB board. That's also going to be impacted based on the location. You might have to have multiple IRB approvals depending on where you're going to be testing the therapeutic. Again, I'm very open to talking about this with anybody who has any kind of questions on any parts of this. So in terms of what our actual timeline looks like with all of the testing. So and this has been from our past experience in rare disease, we've already done this once. So essentially we take 6 months to generate most of our efficacy data and submit the pre-IND package in the same time frame. Again, efficacy can be done for longer than that. This is just the particular time frame we've done previously. So then you submit the pre-IND package to begin talking to the FDA and you determine your clinical trial testing sites. Within about a year, we hope to have toxicology, pharmacokinetics, and biodistribution data all completed and receive our IRB board approval and submit our IND and receive approval. And then we suspect about 18 months till we can actually initiate the phase 1 trial to get all of our clinical testing sites ready. So the cost breakdown, as some of you saw yesterday, and I know our cost is a lot lower than other people. You can talk to me secondary about how I managed to do that, how we managed to do that. So our preclinical costs only average about $400,000 to do most of this testing. Again, because I can do everything in-house. If you need to outsource, that's where you get into the insanity of how much pharmaceutical industry costs. If I have to outsource to a contract manufacturing organization, a contract research organization, my prices are going to quintuple overnight. So that's how we keep our costs down, is I'm able to do everything in-house. In terms of clinical costs, from our past experiences and from a lot of our advisors, this is what a phase 1 roughly costs for us. It's about $30,000 per patient in terms of patient costs, so it runs about $1 million to get through a phase 1. That includes our therapeutic creation costs, which, depending on the dosing regimen, the half-life of the protein, can be more or less based on how much I have to make. So that's kind of our breakdown. So it's about a total of $1.4 million to get us through the initial phase 1. So moving forward, we are ready to go. We've done this previously. We can get into clinic quickly, and the therapeutics are ready to undergo testing. What I ask of the scientific community is all of the input in the world. I'd love to hear your thoughts on what other testing you'd love to see efficacy-wise, and what models you think are best. And that's about what our time frame and plan is. So thank you so much for listening to me.